Okay, hi guys. So today we're going to be doing an overview of identification of cytosolic factors required for nuclear location sequence-mediated binding to the nuclear envelope. This paper is by Adam and Adam and was published in 1994. Okay, so why is this paper important to us? This is our overview as well. Macromolecular movement between cytoplasm and the nucleus is mediated by large structures in the nuclear envelope known as nuclear pore complexes. These are regulated by selective mediated mechanisms, and these regulate the transport of proteins and RNA through this pore complex. This concept was reviewed and supported by many previous studies. There's two classes of amino sequences that localize proteins to the nucleus, and these are known as nuclear localization sequences, or NLS. The first class is characterized by the SV40 large T antigen NLS and consists of 3 to 5 basic residues. The second class of sequences is characterized by the Xenopus nucleoplasm NLS and consists of 2 basic regions of 3 to 4 residues separated by a 10 amino acid spacer. This paper really focuses on the importance in nuclear localization, the role of the proteins that go into nuclear localization are still unclear. A variety of affinity techniques have been identified, cytoplasmic and nuclear NLS binding proteins from a number of sources, and both pore complex and cytoplasmic proteins are required for nuclear transport. Protein transport into the nucleus can be divided into 2 steps. The first step is independent of ATP and temperature, while the second requires ATP hydrolysis. And in this research paper, in order to understand the mechanisms of protein accumulation in the nucleus, they purified and observed soluble cytosolic factors required for transport and studied their relationships. This paper is important to us as a class because we just started talking about the nucleus and nuclear pore complexes. Okay, so for this paper, the methods were cell culture, in vitro nuclear envelope binding reaction, purification of binding components, NEM treatment of purified proteins, and wheat germ agglutinin inhibition of envelope binding. So for the cells, they use Madden-Darby bovine kidney cells. And then for in vitro nuclear envelope binding reaction, permeabilization of MDBK cells were grown on cover slips. And for the binding reaction, the cover slips were prepared and incubated, and a standard reaction was run. In order to evaluate these results, there was photographs taken with a specific camera. In purification of the binding components, NLS receptor was purified from bovine erythrocytes, and nuclear envelope binding activity was purified by the same method. For the NEM treatment of purified proteins, purified NLS receptor and the 97-KD protein were diluted and incubated. Then the proteins were dialyzed and prepared further before application to the permeabilized cells. For the wheat germ agglutinin inhibition of envelope binding, to assess the effects of wheat germ, the cells were first incubated with the wheat germ and then processed for microscopy. Peptides were also used in the study and were obtained from multiple peptide systems and were analyzed by STS-PH. Okay, so on to our results and our figures. Figure 1 here is showing binding to the nuclear envelope. Purified proteins were combined with APC NLS. This assay revealed column fractions that alone caused distribution of APC NLS to the surface of the nucleus. In the top 2 pictures here, we're looking at the equatorial plane of the nucleus, and this shows a discontinuous peripheral nuclear stain. When focused on these specific sections, the fluorescent signal was concentrated at the nuclear periphery with some binding observed in some experiments, but you can see that it's more in the center here at the top. If you look at the bottom 2 pictures, the pattern is more modeled in appearance. On the surface of the nucleus, this fluorescence was discontinuous, similar to the pattern of staining obtained with antibodies to nucleoporins. These results suggested that binding is at the nuclear pore. Here we have figure 4. Figure 4 was showing the ATP and temperature dependence of binding. So as you can see, at 0 degrees Celsius in these two appeared, this one is without ATP and this one's with ATP. Incubation with the purified binding factors at this temperature restricted accumulation of APC-NLS to the nuclear envelope. But with addition of ATP, it resulted in a small amount of internalization, as you can see in some of them with the little dots inside, but there's still weak fluorescence in the nucleoli. With 30 degrees Celsius, without ATP added, there was an increased accumulation in the nuclear interior, and the nuclear envelope fluorescence decreased, however, in comparison to the 0 degrees. These results suggest that the association of NLS containing proteins with the nuclear pore is independent of temperature or the energy state of the cell. The nuclear envelope binding seen with the purified proteins is also not ATP or temperature dependent. Okay, and here we have figure 5, and this is showing that binding requires both P97 and the NLS receptor. As you can see, this first square here is just P97, and you see no binding. And all same with the receptor, you see no binding, but when they are mixed, envelope binding is only seen when both the NLS receptor and P97 are present at the same time. It is likely that this binding is mediated by specific NLS binding proteins, but there is very little evidence for this. And results presented in this figure show that the receptor alone did not lead to the binding of APC-NLS at the nuclear envelope. Okay, on to our results and discussion. So some of the specific results that were gained had to do with the identification of nuclear envelope binding factors. And so that had to do with figure 1 that we talked about, and P97 eluted from the NLS addition anion exchange columns along with envelope binding activity, showed that P97 was the only protein that correlated with receptor-dependent envelope activity. This is very important for the identification of the active species. Envelope binding in each column fraction depended upon the amount of receptor co-purifying with P97, as well as the ratio of the two proteins and the total amount of actors in the assay. Once the receptor was separated from P97, activity was dependent upon independently purified receptor. No other envelope binding activities, receptor-dependent or independent, were observed in any other fractions throughout the purification. Okay, the specificity of binding was also discussed in the results, and the functional wild-type sequence decreased binding by 75%, while the mutant sequence reduced binding by only 21%. These results are in agreement with previous data. ATP temperature and receptor dependence was also discussed, what we saw in figure 4. The association of NLS containing proteins with the nuclear pore is independent of temperature and energy state of the cell, and the binding of APC-NLS at the nuclear envelope is only present when both the receptor and P97 are present. NEM sensitivity of envelope binding was also discussed, and with the modification of P97 with NEM, it decreases binding to the pore, but actually the NEM treatment of the NLS receptor does not decrease the binding. When P97 was treated and mixed with the untreated receptor, binding to the envelope decreased by 75%, and when the receptor was treated and mixed with untreated P97, a smaller decrease of only 27% was shown. NEM treatment of both the receptor and P97 reduced binding to the same level as the treatment of just the P97 by itself. And then they discussed the wheat germ. Nuclear envelope binding is actually inhibited by wheat germ. Agglutinin, this suggests a possible mechanism for the inhibition of transport by lectin, actually, and this takes us to further studies that can be done. Many studies can actually be done to further this research. Looking into cytoplasmic factors in the accumulation of nuclear proteins is super important. The role of these factors are known, but the relationship between them is not. So in the future, papers did look into, researchers did look into the relationships between these factors. And the mechanism of binding to nuclear pore and inhibition by wheat germ can also provide further knowledge into the aspects of the nuclear pore. Thank you for listening.